The human immunodeficiency virus, or HIV, infects cells of the human immune system. Its primary target is a subset of lymphocytes called helper T cells, or CD4 plus T cells, so named because of the CD4 proteins that stud their surface. Proteins on the outer envelope of the virus recognize and bind to the CD4 protein and a co-receptor. When the cellular and viral proteins bind, the cell membrane begins to fuse with the viral envelope. The viral envelope is left at the cell surface. This fusion allows the contents of the viral envelope, the viral core, to enter the cell. Cellular enzymes degrade the viral core proteins, leaving a few viral molecules in the cell cytoplasm, including the single-stranded RNA genome of the virus. Several viral enzymes, including reverse transcriptase, also remain in the cytoplasm. HIV is a retrovirus, which means that it uses RNA molecules rather than DNA as its genetic material. Using reverse transcriptase, it makes a DNA copy of the two single-stranded RNA molecules it carried into the cell. Reverse transcriptase also has two other enzymatic activities. With an RNase activity, it degrades the RNA. With a DNA polymerase activity, it makes a second strand of DNA. The resulting double-stranded DNA is transported into the cell nucleus through the nuclear pores and is inserted into the host's chromosomal DNA. The host RNA polymerase may transcribe the viral DNA at relatively low rates for several months or even years. However, if the T cell is activated by any of several factors in the blood, the cell's transcriptional machinery increases the rate of transcription of viral DNA into messenger RNA. Some of the RNA molecules are processed, resulting in a variety of RNA transcripts. These RNA molecules enter the cytoplasm, where the cell's own ribosomes translate the mRNA into proteins. Some of the full-length mRNA transcripts will serve as new viral genomes. Ribosomes on the endoplasmic reticulum translate the RNA molecules that contain sequences for viral envelope proteins. These proteins are transported to the Golgi apparatus, where they are processed. The proteins then move to the plasma membrane and are embedded there. Free ribosomes translate other viral mRNAs. In the case of the structural proteins, a single long polypeptide is produced. Protease, another viral enzyme, cuts the polypeptide into distinct proteins. In this way, the cell's machinery produces many copies of both the viral proteins and the full-length viral mRNA molecules. Viral mRNA molecules and viral proteins, including reverse transcriptase and structural proteins, are assembled into a viral particle that buds from the host cell membrane. As it buds, it takes a portion of the cell membrane and the embedded envelope proteins with it. A cell produces many viral particles during an infection, but ultimately the cell will die.